Good morning students. We'll start our lecture 4 of fundamental course with today's topic on plant growth regulators. What are plant growth regulators? It is a substance which decides or which controls the growth of the plant, which play an important role to grow faster, and the development process of the plant is dependent on these growth regulators. So plant growth regulators is also called as phytohormones, are chemicals, that substance that influences the growth of a plant. So, and the differentiation of plant cells, tissue and organ. So, plant growth regulator function as a chemical messenger. This term is important. It acts as a chemical messenger for intercellular communication, and they work together coordinating the growth and development of a cell. So, this growth regulator plays a very important role in coordinating the growth and development of the cell. In last class, we spoke about the growth of the plant, the development of the plant, the photorespiration, all these. So growth regulators will coordinate the growth and development of the plant. So they could be indole compounds such as, so the growth regulator, indole acetic acid, indole compound, or adenine derivatives such as N6 furfuryl aminopurines, or kinetine derivatives of carotenoids such as abscisic acid. Then we have terpene derivatives such as gibberellic acid, or gaseous derivatives such as ethylene. So remember, the growth regulators can be indole derivatives such as indole acetic acid, indole butyric acid. It can be adenine derivative such as adenyl purines, or it is a carotenoid derivative such as abscisic acid, or it is an terpene derivative such as gibberellin, or gaseous derivative such as ethylene. So the classification of plant growth regulator is done into two groups: the growth promoters and the growth inhibitors. What are growth promoters? We have three good examples of growth promoters: Auxins, Gibberellins, and Cytokinins. And the growth inhibitors are abscisic acid and ethylene. Remember as a code: AGC, or ACG, Agriculture College, Gulbarga, as Auxins, Gibberellins, Cytokinins are growth promoters, and abscisic acid, ethylene are the growth inhibitors. First, starting with the growth regulator, the growth promoter, Auxins. It was developed by or discovered by Charles Darwin and Francis Darwin. It was Charles Darwin and Francis Darwin discovered the Auxin, and F.W. Went isolated Auxins from the tip and coleoptile of oat seedlings, and Went developed the Avena curvature test for the bioassay of Auxins. Remember, Auxin was first discovered by Darwin, but it was first isolated by F.W. Went from the tips of coleoptile of oat seedling, and Went developed the Avena curvature test to test the bioassay of Auxins, and which are the synthetic, which are not naturally available, synthetic Auxins such as indole acetic acid, indole butyric acid, naphthalene acetic acid, 2,4-D, 2,4-dichlorophenoxyacetic acid. These 2,4-D is also an herbicide. So these are the synthetic Auxins. What is the physiological effect of Auxin, or what is the role of Auxin? The first role is promote the apical dominance. This is the plant, the apical portion of the plant. It plays an important role in dominance of the apical growth. So apical dominance initiate the rooting in the stem cuttings. So whenever you go for cutting sowing, just you have to cutting planting, you have to dip it in the Auxin solution, which promotes the growth of the plant. It promotes the flowering in case of pineapple. It prevents the fruit and leaf drop at early stages. So it is a growth regulator. So it promotes the abscission of older mature leaves. It induces the parthenocarpy. This is important. Production of seeds without fertilization. It induces the parthenocarpy in tomatoes, and it uses as a herbicide, for example 2,4-D. So it controls xylem differentiation in the and helps in the cell division. The next, after the first growth promoter, Auxin, will go with the second growth promoter, cytokinin. So cytokinin was developed by Skung and Miller, discovered the cytokinin. Auxin, F.W. isolated, where Skung and Miller discovered the cytokinin. and they crystallize the cytokinin, promoting the active substance named as kinetin from the corn kernels and coconut milk. From the cytokinin, they produce the active substance called as kinetin. Kinetin. Remember the term kinetin. It was Coombe and Miller developed the cytokinin. What is the physiological effect of cytokinin? It induces the cell division, plays a very important role in cell division, delay the senescence, means aging of the leaves and other organs. It increases the cell expansion in a dicotyledons and leaves and promotes the chloroplast development and chlorophyll synthesis. It plays an important role in chloroplast development and chlorophyll synthesis. The next is the third growth promoter, gibberellin. It was developed by Kurosawa, Japanese scientist, identified gibberellin present in a fungal pathogen Gibberella fujikuroi. From a Gibberella fujikuroi, he identified the gibberellin. The gibberellin were isolated from the infected rice seedling treated with a sterile filtrate of fungus. So the gibberellin was isolated by Kurosawa from a pathogen Gibberella fujikuroi. The gibberella were isolated from an infected rice seedling when treated with the sterile filtrates of fungus. And the physiological effect of these gibberellin is that it plays a very important role in elongation of intact stem, dark shoots, and bolting in the rosette plants such as cabbage, overcoming the natural dormancy. So the gibberellins are also called as dormancy breaker. So it is overcoming the natural dormancy of buds, tubers, seeds, and it induces the parthenocarpy or development of seedless food. That is development of seedless fruit such as banana. I expressed in the wrong way. Auxins, parthenocarpy means development of fruit without fertilization, without seeds. So they promote flowering in a long day plants and vernalization. It plays a very important role in flowering under cold temperature, vernalization. So we discussed in last class. So promote flowering in long day plant. It induces the parthenocarpy and development of the seedling fruit. These are the advantages of gibberellin. So after learning the growth promoter, we will deal about growth regulator or growth regulator such as ethylene. It was Auxins confirmed that release of volatile substance from the ripened orange that hastened the ripening of stored unripened banana. So the volatile substance was identified as ethylene. It was the Auxin. He kept those oranges in a banana room where banana was stored, and banana riped faster as compared to that of a normal day. So they confirmed it as a ethylene. The physiological effect of cytokinin is that the physiological effect of ethylene promote the senescence and abscission, induces the fruit ripening, it breaks the seed and bud dormancy, initiates the germination in peanut seeds, sprouting of potato tubers, and promote the root growth and root hair formation. These are the advantages of ethylene. Then another growth regulator is abscisic acid. Abscisic acid was developed by Frederick Adicott and co-workers, identified the abscisic acid, which was isolated from several abscission accelerating substance from the cotton plant. And abscisic acid is also called as stress hormone, very important for the competitive exam point of view. Abscisic acid is also called as stress hormone. The physiological effect of cytokinin acts as a general plant growth regulator. It induces the seed and bud dormancy. Inhibits the seed germination. It stimulates the closure of stomata, and it plays an important role in seed development, maturation, and dormancy. So these all are the important roles of a abscisic acid. That's a stress regulator, a stress hormone, releases the stress hormone, which play a very important role in closing of stomata, bud dormancy, plant growth inhibitor, and it plays important role in seed development and maturation. So generally under maturate stage, we see this hormone which plays a very important role in growth and development. If you are liking my teaching, please comment below. Thank you.